Welcome back. We finished up our discussion of glycolysis with the pyruvate molecule. Now we want to consider what might happen to that pyruvate molecule. Now, when I say what might happen, you want to realize that at this point that I get to pyruvate, there are various options provided to different kinds of living things. We're going to take an option where we look at what happens to pyruvate if it continues through the process of respiration. But we're going to come back to this point again in the future and consider what happens to pyruvate if it's sent through what's called a fermentative pathway. So remember this, once we get to pyruvate, different options that different kinds of cells have. Let's take a look at what happens to pyruvate if it continues through the respiratory pathway. Well, in terms of the overview of our process of respiration, you can see where we've moved to now. We've completed glycolysis, and so now we're going to be looking at this event here, the next component of respiration. We're going to be primarily focusing on a sequence of chemical reactions called the Krebs cycle, or called the Citric Acid Cycle, or microbiologists like to refer to it as the Tricarboxylic Acid Cycle, TCA cycle. Different names, same process. Krebs is the name of the individual who first discovered this and figured out the steps involved. You can also see that we're now inside of the mitochondria of the cell. If we're dealing with a eukaryotic cell, the pyruvate molecule has now moved inside of the mitochondria. If this were a prokaryotic cell, a bacterial cell, we'd be still carrying this out in the cytoplasm of the cell. All right, so let's consider the overview illustration that I've provided to you. Make sure you find this one. This should be the second page of the set of illustrations that are the metabolism illustrations. We're going to take this apart and look at the individual components. All right, so what are we going to do? First of all, we have to take that pyruvate molecule, and in a sense, we have to get the pyruvate ready to be added to the molecules of the Krebs cycle. Pyruvate itself can't move right into the Krebs cycle, so there's what some people like to call a get-ready step or a preparatory step. I take the pyruvate and I'm going to turn it into a molecule called acetyl. And then I'm going to bind the acetyl molecule to another molecule called coenzyme A. So in a summary view, we could say we're taking pyruvate and we're turning it into acetyl coenzyme A. Notice a couple things that happen during this sequence of events. We're going to make some NADH, and we're also going to release some CO2. So we're going to make sure we keep track of these things. All right? Take a look at the illustration I provided to you. We have our pyruvate molecule, and notice we have two pyruvates, don't we? If we're going to keep track of everything that happens to that original glucose, we got to think about what's happening to two pyruvates at this point. Remember, each pyruvate is a 3-carbon chain. We're going to take that pyruvate molecule and turn it into an acetyl molecule. An acetyl molecule is a 2-carbon chain. That means right away that I should be looking for some carbon being released. And notice, from this event, from this part of the process, there is indeed some CO2 released. That's 2 of the carbons. That's those carbons that were part of the pyruvate, carbons that were part of the original glucose, right? Okay, so that's part of the waste material that's being released. Also notice that I take some NADs and turn them into NADHs. For each pyruvate that gets turned into an acetyl molecule, I make 1 NADH. But since I have 2 pyruvates, and I'm trying to keep count of the overall process, we'd say that 2 NADHs get turned into 2 NADs get turned into 2 NADHs. Now, if I had to label some of these arrows, think about what I'd label them. Wouldn't this arrow right here be labeled as reduction? Think about that and see if you agree with my labeling. Also, wouldn't this arrow right here be referred to as oxidation? Okay? Think about what I'm saying. NAD is becoming NADH. It's gaining hydrogens, experiencing reduction. Pyruvate is getting turned into acetyl, and those hydrogens that NAD got had to come from somewhere. They must have come from pyruvate. So we could say that pyruvate experiences oxidation to get turned into the acetyl molecule. By the way, you might also see some books talk about what's happening to pyruvate as decarboxylation. Well, you can see what that name's implying, that the pyruvate is having some carbon removed, decarboxylation. That's the CO2s that are being released. So you might see this referred to in different ways, but in particular, take a look at it and see if it makes sense that it's a redox reaction. NAD being reduced, pyruvate being oxidized. Now, what happens to this acetyl molecule once it's produced? Well, it binds to a molecule called coenzyme A. Coenzyme A, as its name implies, is a coenzyme molecule. It's floating around inside of the interior of the mitochondria, in the matrix of the mitochondria. And it binds to the acetyl molecule. So one acetyl with one coenzyme A makes a molecule called acetyl-CoA, acetyl coenzyme A. Since we have two acetyls, that would happen 2 times. We're going to see in a minute that the coenzyme A is just going to act as a delivery truck. It's going to help the acetyl molecule get into the Krebs cycle, and then the coenzyme A comes back to pick up another acetyl, so it's recycled. So notice that here we see that there's a process where there's coenzyme A being recycled after the acetyl portion gets dropped off at the Krebs cycle. And that's what we want to take a look at next. By the way, notice that during this event, I haven't made any ATPs. So keep that in mind as well. Keep track of that there were no ATPs made directly from this event. Now, the Krebs cycle itself, as the name implies, is a cyclical event. It's a sequence of chemical reactions, which means that the molecule that starts the process is also the molecule that ends up the process. That's why it's called a cyclical event. Notice what we're feeding into this process. We're feeding into it those 2 carbons of the acetyl. Notice what we're producing as a result of this event. We're going to release some CO2. We're going to make some NADH. We're going to make some ATP. And we're going to produce, this is the first time we've seen it, some FADH. FADH2, an electron carrier like NAD. Remember, it carries the same quantity of material as NAD does. All right? So we're going to want to look for this event. Notice, this illustration shows just one acetyl going through the citric acid cycle. But of course, since we know we have 2 acetyl molecules, we're going to want to keep track of what happens to both of those acetyl molecules as each of them goes through their own individual Krebs cycle. So in our illustration, look what I've done. I've tried to summarize the overall event and note the name of a few key molecules. First of all, we have the acetyl molecules coming from the previous step. Already present within the matrix of the mitochondria is going to be a molecule called oxaloacetate. You might also see it called oxaloacetic acid. The acetyl molecule binds to the oxaloacetate. The 2 carbons of acetyl get added to the 4 carbons of oxaloacetate, and I make a 6-carbon molecule, citric acid. That's the reason this is called the citric acid cycle, because the very first molecule produced in the cycle is a molecule called citric acid. It's also called the Krebs cycle because Krebs was the individual who determined the steps involved. And it's called the TCA cycle, especially by microbiologists, the tricarboxylic acid cycle, because throughout this event, there are 3 carboxylic acids involved. Now, I'll either use citric acid cycle or Krebs cycle, or in many cases, I'll use both of those terms simultaneously. Rarely do I use the term TCA cycle, but I might mention it to you just to remind you that it's another name for this same sequence of events. Notice everything is times 2, right? Because we have 2 acetyls. By the time this process is over, guess what we're going to be left with? Oxaloacetate again. And then it'll be ready to start the next cycle. So remember, it's a cyclical event because the molecule that was present to begin the event is the molecule that's present at the end of the event as well. All right, so let's consider what's going on in this process. We have these 2 acetyls binding to 2 oxaloacetates to make my 2 citric acid or citrate molecules. Sequence of chemical reactions takes place. We'll take a look at some of those in a little more detail in just a moment. But this summary is what I'd like you to know about. Notice what I've done. I've indicated to you the materials produced in this column over here. I also have for you what necessary raw materials would have to be present for these products to be made. Note one other thing. Notice that for each of the boxes I have on the side over here, I've given you the amount of material used for one turn of the cycle, one cycle, and the amount of materials produced in total for 2 turns of the cycle. Why do we want to know that? Remember, we have 2 acetyl molecules. So that means this cycle is going to happen 2 times. Let's take a look at the summary of what we're getting made. We're going to have NADs being reduced to NADH. For each 1 turn of the cycle, that happens 3 times. Collectively, overall, we would have 6 NADHs produced from 2 turns of the cycle. We're going to have a second electron carrier molecule involved. That second electron carrier molecule is FAD. For 1 turn of the cycle, 1 of those gets reduced. For 2 turns of the cycle, 2 get reduced. So we could say that we produce 2 FADHs. We also are going to have an event of substrate level phosphorylation, where energy from one of the substrate molecules in this chemical reaction is going to be used to build an ATP molecule. So we'd expect there to be 1 substrate level phosphorylation event for each cycle. Since we have 2 cycles occurring, we're going to get 2 ATPs from the process. Finally, if you think about it, if we started off with 6 carbons, citric acid, and we end up with only 4 carbons, oxaloacetate, some carbons had to go somewhere, right? So we would expect there to be some CO2 released from this process. And sure enough, we're going to see a total of 4 CO2s produced. Why 4? 2 CO2s for each acetyl that enters the cycle. So 4 CO2s produced overall. All right, you'll want to think about it both in terms of an individual cycle and in particular for the overall production. Remember, we're tracing this all back to that 1 glucose we began with. All right? Let's take a look at another way of representing this process. You can see that this is a little bit more detailed. It doesn't give the names for all the different molecules, but let's add some. Notice we're beginning the process by bringing that acetyl molecule in, the 2 carbons of acetyl. It's being added to oxaloacetate to make citric acid. Now, those were the key molecules I wanted you to know about. So these additional molecules that are going to be labeled on here, you might find interesting to take a look at. But remember, you do not have to know the details of their names or where they fit into the cycle. These 3 molecules that are shown right now, the acetyl, the oxaloacetate, and the citric acid, are the 3 molecules I want you to know about. But let's take a look at some of these other steps involved. Notice what's going to happen. In the next couple steps, I'm going to carry out the reduction of some NAD 2 times. I'm also going to make some ATP, substrate level phosphorylation. And what we're going to expect to find is that there's going to be some CO2 released as well. If we continue on to the end of the process, you can see that I'm going to carry out the reduction of FAD to FADH2. And I'm going to, one more time, carry out the reduction of NAD to NADH. And I'm going to finally end up the process with oxaloacetate again. So again, looking at this more detailed representation may help you understand better the summary level of information I'd like you to know about. Notice I'm not asking you to understand exactly where each of the NADHs is made or where the CO2s are produced or the names of these other molecules in this process. But seeing this illustration may help you understand that summary a little bit more clearly. By the way, notice this represents the information for what? Just 1 turn of the cycle. And remember, you're going to want to know in summary form the total amount of material produced for 2 turns of the cycle. So let's take a look at a metabolic summary of this event. And notice, we're going the whole way back and we're including both the acetyl formation step and the citric acid cycle part of it. We're combining those together as one overall event. Overall, we've made 8 NADHs. You want to make sure you go back and find all of them. 2 of them are formed during that acetyl formation step, 6 more during the citric acid cycle. We've made 2 FADHs, 2 FADH2s. We've also made 6 CO2s. You'll want to find those. Find where those 6 carbons are being released. 2 from acetyl formation, 4 from the citric acid cycle. We have ATP produced, substrate level phosphorylation. And make note for yourself that that only happened during the citric acid cycle. If you think about it, we've completed the breakdown. We've completed the decomposition of our original glucose molecule, haven't we? Because we've taken all of those carbons that used to be together in the 6-carbon chain of glucose, and now they're separate CO2s. We've completely broken apart that chain. Excuse me. We've also, during this process, loaded more of the electrons, hydrogen atoms from glucose, the original glucose, onto electron carriers, NAD, FAD, so that we have FADH2 and NADH. Remember, eventually we're going to see those hydrogen atoms, those electrons that they're carrying, dumped somewhere, electron transport chain. And of course, that will be one of our future topics. All right, go back and review this event. Make sure you think of it as a combined event. Think about where it's occurring in a eukaryotic cell versus a prokaryotic cell. Okay? So, the 2 key events, making the acetyl molecule, using it along with oxaloacetate to drive the citric acid cycle, make citrate, were events that we think of as being the completion of the breakdown of the carbohydrate molecule. But take a moment to think about something. If you go back and you check, how many ATPs have I made so far? From glycolysis, from acetyl formation in the citric acid cycle, well, geez, I've only made net 4 ATPs so far. But I thought that I was going to get a lot more ATPs out of that glucose molecule. I thought I was going to get 36 or 38 ATPs. Well, that must mean that the last third of this process, the electron transport process, the chemiosmotic event that we're going to talk about next time, that must be where the big energy payoff is, right? Well, how could that be? Well, all you have to do is take a moment to count up the electron carriers that are present at this point. If you go back and check all your different parts of the illustration, you should be able to find 10 NADHs. Remember, each one of those NADHs is like a little package of energy. It's carrying excited electrons, high energy electrons. Also, you're going to find 2 FADHs, aren't you? Other electron carriers. So that means that the next event, the electron transport chain, is going to have as a key component of it how we're going to make use of those electron carriers to complete our production of ATP. I look forward to talking to you about that very interesting event next time.